Enzyme. So enzyme is all enzymes are protein, but all proteins are not enzyme. This is the statement you must have heard. So human body is composed of different type of the cells, tissues and other complex organ, and for the efficient functioning our body releases some chemicals. Okay, some chemicals to accelerate the biological processes such as the respiration, the digestion, the excretion, and also few metabolic activities to sustain a healthy life. Hence, these chemicals or these protein are enzyme only which can accelerate the biochemical reaction in our body. So hence we can say that enzymes are very pivotal in all the living entities which governs all the biological process. So we can define the enzyme that it is a substance that act as a catalyst. Catalyst means that molecule or any compound that can accelerate the chemical reaction. So enzyme is a substance that acts as a catalyst in the living organisms, regulating the rate at which the chemical reactions proceed without itself being altered in the process. Means these enzymes they use to, suppose this is a substrate and with the help of the enzyme it is converted into the product. So in this whole journey from converting from the substrate to the product, this enzyme does not get altered. This remain as it is, and once the product has been formed, this enzyme is then prepared for the other substrate to get attached so that it can convert that substrate also into the another product. So let's see or figure out what are the different characteristics of these enzymes. So enzymes are typically we know are the proteins only that act as a biological catalyst, but some nucleic acid they also have some catalytic activity, and these nucleic acid are known as the ribozyme. Okay. Next, enzyme structure. The enzyme is just like the protein because they all are proteins. So they have the primary, secondary, tertiary, and the tertiary structure. Okay, and the tertiary structures only involves the foldings of the protein chain upon itself, thus creating some crevices like this. Okay, suppose this is an enzyme, so some crevices or some pocket has been created so that the substrate can attach themselves to that void spaces. Okay, the active site, and the active site is just a pocket or the crevices, this one only in the enzyme. Okay, this is known as the active site, this one. So it is a pocket or crevices in the enzyme's tertiary structure where the substrate used to bind. So the substrate used to come and it get bind itself in the active sites. So these active sites are very important for the enzyme because it is crucial for catalyzing the reactions at a very high rate. Enzyme catalyst versus the inorganic catalyst. The enzyme catalyst is differ from the inorganic catalyst in the various way. Inorganic catalyst basically they work at a very high temperature and the high pressure. Okay, so they used to work at very high temperature and high pressure, while enzymes they are sensitive to the high temperature, that is above the 400 degrees Celsius. They are very sensitive to temperature. So this is the difference between the enzyme catalyst and the inorganic catalyst. The thermal stability. Obviously, the enzyme are very much sensitive to the temperature. So, enzyme which are isolated from the organism, which are living, who are living in the extreme conditions such as the high temperature, for example, the hot wind and the sulfur spring. So, they can be stable and they retain the catalytic activity or the power at even a very high temperature. So thermal stability can be possible in the extreme if they are being isolated or extracted from the organism who are living at a very high temperature or the extreme condition. So thermal stability is very important quality of the enzyme from the thermophilic organisms. Thermophilic organism means those living at a very high temperature. So the damaged enzyme, generally they get damaged at a high temperature, which can lead to the denaturation. Means the nature of the enzyme gets denatured and the loss of the catalytic activity takes place. So the importance of these active site is that it is the key reason only where the substrate binding and the catalysis occur. If the catalysis or the active site is not active, then the rate of acceleration gets slowed down, and also the various other various other things get affected to the enzyme. So the enzyme accelerate the reaction by providing the alternative pathway with the lower activation energy. Okay. So specificity and the catalytic power, obviously the enzyme they exhibit the substrate specificity, means the specific substrate only get attached to the active site. It's not like suppose this is the enzyme and the active site is this, and suppose there are different shapes of the substrate are present. Some are spherical, some are rhomboid shape, hexagonal shape, some are pentagon shape type structure. So which substrate used to bind that particular enzyme? So obviously this substrate, because it get itself attached in a very we can say in a very fixed manner to the enzyme. So the enzyme they always exhibit the substrate specificity, recognizing and binding to the specific substrate. Enzyme catalyzes the reaction at a very high rate, making the biological process efficient. So let's talk about what are the chemical reactions. So chemical reaction are obviously of the two type, the physical chemical reaction and the chemical reaction. So physical chemical reaction is just that it is occurring in the change in the shape of the substrate without breaking the bond. For example, the ice is converted into the water. Okay, means it comes the component which have in the ice is same as in the water, just the form or the shape has been changed. While the chemical reaction, it involves the breaking and the forming of the bond during the transformation. So these are two type of the